Chapter 1 Introduction to Clinical Trials. Outlines. Introduction to Clinical Trials. Fundamental point. What's a clinical trial? Clinical trial phases. Preclinical investigations. Phase 1 studies. Phase 2 studies. Phase 3 studies. Phase 4 trials. Introduction to clinical trials. The evolution of the modern clinical trial dates back to the 18th century. Lind, in his classical study on board the Salisbury, evaluated 6 treatments for scurvy in 12 patients. One of the 2 who was given oranges and lemons recovered quickly and was fit for duty after 6 days. The second was the best recovered of the others and was assigned the role of nurse to the remaining 10 patients. The concept of randomization was introduced by Fisher and applied in agricultural research in 1926. The first clinical trial that used a form of random assignment of participants to study groups was reported in 1931 by Ampersand et al. After careful matching of 24 patients with pulmonary tuberculosis into comparable groups of 12 each, a flip of a coin determined which group received sanocrizin, a gold compound commonly used at that time. The British Medical Research Council trial of streptomycin in patients with tuberculosis, reported in 1948, was the first to use the random numbers in the allocation of individual participants to experimental and control groups. The principle of blindness was also introduced in the trial by Ampersand et al. The participants were not aware of whether they received intravenous injections of sanocrizin or distilled water. In a trial of cold vaccines in 1938, Dale and co-workers referred to the saline solution given to the subjects in the control group as placebo. It's only in the past several decades that the clinical trial has emerged as the preferred method in the evaluation of medical interventions. Fundamental point. A properly planned and executed clinical trial is a powerful experimental technique for assessing the effectiveness of an intervention. What's a clinical trial? We define a clinical trial as a prospective study comparing the effect and the value of interventions against a control in human beings. Note that a clinical trial is prospective rather than retrospective. Study participants must be followed forward in time. They need not all but followed from an identical calendar date. In fact, this will occur only rarely. Each participant, however, must be followed from a well-defined point in time, which becomes time zero or baseline for the study. A clinical trial must employ one or more intervention techniques. These may be single or combinations of diagnostic, preventive, or therapeutic drugs, biologics, devices, regimens, or procedures. Intervention techniques should be applied to participants in a standard fashion in an effort to change some aspect. Early phase studies may be controlled or uncontrolled. Although common terminology refers to phase 1 and phase 2 trials because they are sometimes uncontrolled, we will refer to them as clinical studies. A trial, using our definition, contains a control group against which the intervention group is compared. At baseline, the control group must be sufficiently similar in relevant respects to the intervention group in order that differences in outcome may reasonably be attributed to the action of the intervention. Most often, a new intervention is compared with or used along with best current standard therapy. Only if no such standard exists or for several reasons is not available, is it appropriate for the participants in the intervention group to be compared to participants who are on no active treatment. No active treatment means that the participant may receive either a placebo or no treatment at all. Obviously, participants in all groups may be on a variety of additional therapies and regimens, so-called concomitant treatments, which may be either self-administered or prescribed by others, for example, personal physicians. Equally important is the need for and responsibility of the investigator to fully inform potential participants about the trial, including information about potential benefits, harms, and treatment alternatives. Unlike animal studies, in clinical trials, the investigator cannot dictate what an individual should do. He can only strongly encourage participants to avoid certain medications or procedures which might interfere with the trial. Since it may be impossible to have pure intervention and control groups, an investigator may be not able to compare interventions, but only intervention strategies. Strategies refer to attempts at getting all participants to adhere, to the best of their ability, to their originally assigned intervention. When planning a trial, the investigator should recognize the difficulties inherent in studies with human subjects, and attempt to estimate the magnitude of participants' failure to adhere strictly to the protocol. The ideal clinical trial is one that is randomized and double-blind. Deviation from this standard has potential drawbacks. In some clinical trials, compromise is inevitable, but often deficiencies can be prevented by adhering to fundamental features of design, conduct, and analysis. Efficacy or explanatory trials refer to what the intervention accomplishes in an ideal setting. Effectiveness or pragmatic trials refer to what the intervention accomplishes in actual practice, taking into account incomplete adherence to the protocol. Clinical trial phases. While we focus on the design and analysis of randomized trials comparing the effectiveness of two or more interventions, several steps or phases of clinical research must occur before this comparison can be implemented. Classically, trials of pharmaceutical agents have been divided into phases from 1 to 4. Studies with other kinds of interventions, particularly those involving behavior or lifestyle change or surgical approaches, will often not fit neatly into those phases. In addition, even trials of drugs may not fit into a single phase. For example, some may blend from phase 1 to phase 2 or from phase 2 to phase 3. Therefore, it may be easier to think of early phase studies and late phase studies. Nevertheless, because they are still in common use and because early phase studies, even if uncontrolled, may provide information essential for the conduct of late phase trials. The phases are defined below. Preclinical investigations. Preclinical investigations include animal studies and evaluations of drug production and purity. Animal studies explore the drug's safety in doses equivalent to approximated human exposures, pharmacodynamics, including mechanisms of action and the relationship between drug levels and the clinical response, and the pharmacokinetics, including drug absorption, distribution, metabolism, excretion, and potential drug-drug interaction. Phase 1 studies. Although useful preclinical information may be obtained from in vitro studies or animal models, early data must be obtained in humans. People who participate in Phase 1 studies generally are healthy volunteers, but may also be patients who have typically already tried and failed to improve on the existing standard therapies. Phase 1 studies attempt to estimate tolerability and characterize pharmacokinetics and pharmacodynamics. They focus on questions such as bioavailability and body compartment distribution. They also provide preliminary assessment of drug activity. NP bioavailability is the amount of drug which reaches the site of physiological activity after administration. Phase 1 studies. Often, one of the first steps in evaluating drugs is to estimate how large a dose can be given before unacceptable toxicity is experienced by participants. This dose is usually referred to as the maximally tolerated dose. Much of the early literature has discussed how to extrapolate animal model data to the starting dose in humans, or how to step up the dose levels to achieve the maximally tolerated dose. In estimating the maximally tolerated dose, the investigator usually starts with a very low dose and escalates the dose until a pre-specified level of toxicity is obtained. Typically, a small number of participants, usually 3, are entered sequentially at a particular dose. If no specified level of toxicity is observed, the next predefined higher dose level is used. If unacceptable toxicity is observed in any of 3 participants, an additional number of participants, usually 3, are treated at the same dose. If no further toxicity is seen, the dose is escalated to the next higher dose. If an additional unacceptable toxicity is observed, then the dose escalation is terminated and that dose, or perhaps the previous dose, is declared to be the maximally tolerated dose. This particular design assumes that the maximally tolerated dose occurs when approximately 1/3 of the participants experience unacceptable toxicity. Variations of this design exist, but most are similar. Some have proposed more sophisticated designs in cancer research that specify a sampling scheme for dose escalation and statistical model for the estimate of the maximally tolerated dose and its standard error. The sampling scheme must be conservative in dose escalation so as not to overshoot the maximally tolerated dose by very much, but at the same time be efficient in the number of participants studied. MB, often people with different types of cancer can take part in the same Phase 1 study. Phase 1 studies, step up, step down. Many of the proposed schemes utilize a step up, step down approach, the simplest being an extension of the previously mentioned design to allow step downs instead of termination after unacceptable toxicity, with the possibility of subsequent step-ups. Further increase or decrease in the dose level depends on whether or not toxicity is observed at a given dose. Dose escalation stops when the process seems to have converged around a particular dose level. Phase 1 studies, Bayesian, The phrase Bayesian statistics refers to an approach and method of analysis which combines prior knowledge and accumulated experience with current information in order to make inferences about a quantity of interest. Bayesian approaches have also been developed. These involve methods employing continual reassessment and escalation with overdose control. Bayesian methods involve the specification of the investigator's prior opinion about the agent's dose toxicity profile, which is then used to select starting doses and escalation rules. The most common Bayesian Phase 1 design is called the continual reassessment method, in which the starting dose is set to the prior estimate of the maximally tolerated dose. After the first cohort of participants, typically of size 1 or 2 or 3, though other numbers are possible, the estimate is updated and the next participants assigned to that estimate. The process is repeated until a pre-specified number of participants have been assigned. The dose at which a hypothetical additional participant would be assigned constitutes the final estimate of the maximally tolerated dose. Bayesian methods that constrain the number of total toxicities have also been developed, escalation with overdose control, and have designs that allow for 2 or more treatments, and as have methods that allow for incomplete follow-up of long-term toxicities. Time to event continual reassessment method. Many variations have been proposed. An advantage of Bayesian Phase 1 designs is that they are very flexible, allowing risk factors and other sources of information to be incorporated into escalation decisions. A disadvantage is their complexity, leading to unintuitive dose assignment rules. Phase 2 studies, also referred to as therapeutic exploratory trials, are usually larger than Phase 1 studies and are conducted in a small number of volunteers who have the disease of interest. They are designed to test safety, pharmacokinetics, and pharmacodynamics, but may also be designed to answer questions essential to the planning of Phase 3 trials, including determination of optimal doses, dose frequencies, administration routes, and endpoints. They may offer preliminary evidence of drug efficacy by comparing the study drug with historical controls from published case series or trials that established the efficacy of standard therapies. 2. Examining different dosing arms within the trial. Or 3. Randomizing subjects to different arms, such as control arm. However, the small number of participants and primary safety concerns within a Phase 2 trial usually limit its power to establish efficacy and thereby support the necessity of subsequent Phase 3 trial. MB arm is a group or subgroup of participants in a clinical trial that receives a specific intervention or treatment or no intervention according to the trial's protocol. One of the traditional Phase 2 designs in cancer is based on the work of Sheehan, which is a version of a two-stage design. In the first stage, the investigator attempts to rule out drugs which have no or little biologic activity. For example, He may specify that a drug must have some minimal level of activity, say in 20% of participants. If the estimated activity level is less than 20%, he chooses not to consider this drug further, at least not at that maximally tolerated dose. If the estimated activity level exceeds 20%, he will add more participants to get a better estimate of the response rate. A typical study for ruling out a 20% or lower response rate enters 14 participants. If no response is observed in the first 14 participants, the drug is considered not likely to have a 20% or higher activity level. The number of additional participants added depends on the degree of recession desired, but ranges from 10 to 20. Thus, a typical cancer phase 2 study might include fewer than 30 people to estimate the response rate. The precision of the estimated response rate is important in the design of the controlled trial. In general, phase 2 studies are smaller than they ought to be. Phase 3 trials, also referred to as a therapeutic confirmatory or comparative efficacy or pivotal trial, may be pursued. This stage of drug assessment is conducted in a larger and often more diverse target population in order to demonstrate and confirm efficacy and to identify and estimate the incidence of common adverse reactions. However, given that phase 3 trials are usually no longer than 300 to 3,000 subjects. Phase 3 trials. The most common type of phase 3 trials, comparative efficacy trials, often referred to as superiority or placebo-controlled trials, compare the intervention of interest with either a standard therapy or a placebo. Another type of phase 3 trial, the equivalency trial or positive controlled study, is designed to ascertain whether the experimental treatment is similar to the chosen comparator within some margin pre-specified by the investigator. Phase 4 trials, also referred to as therapeutic use or post-marketing studies, are observational studies performed on FDA-approved drugs to identify less common adverse reactions and evaluate cost and drug effectiveness in disease populations or doses similar to or markedly different from the original study population.